Okay, so now we're going to discuss translation. First, we have to familiarize ourselves with what a genetic code is. So, the genetic code is basically the code that our tRNAs follow to know what type of amino acids they're going to encode. So, for example, if you have your mRNA, let's say AUG, GGC, AC, or something like that. So they are read every three bases, and these three bases, they're called a codon. So that is for the mRNA, whereas the anticodon, which is basically the part that is um, complementary to this, is found in your tRNA. So in the genetic code for a specific sequence, it has a corresponding amino acid. So what are the characteristics of the genetic code? Firstly, the genetic code is non-overlapping, meaning if you have here three bases, you don't read it as AUG and then UGG. No, we read it as AUG, and then after that, we read the next series. So they are a non-overlapping code. Because we have to consider that it is much better for the ribosome for it to be non-overlapping, because then it will allow better movement within the ribosome, as well as it lessens the confusion and inefficiency in the cell. Also, the code is continuous, meaning after each codon, you read the next codon. They have no concept of a pause. Walang stop moment dito. So, meaning if you have your AUG, this codes for something, and then the next one also codes for something. There's no AUG and then G is considered a pause or something. That does not happen. So the genetic code is overlapping, and it is non-overlapping rather, and it is continuous. Also, the genetic code is universal, meaning all species here on Earth so far subscribe to this genetic code. Meaning, if you have a UUU in your mRNA, then that will encode for a phenylalanine. So I would like to emphasize that these UUU, UUC, etc., these are the codes found on the mRNA. So these are mRNA codons, okay? They are not anticodons of your tRNA. So if you have a table like this part here usually indicates the first position, this one the second, and this is the third. So meaning, if we read it, this is U, 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 and this will give us phenylalanine here. Or, in here you can see C, C, U, and it encodes for proline. So these are the codons found on the mRNA, and these are the amino acids they encode for. So, if you will see in this section here, the ones highlighted in gray, the third base does not contribute much to the difference in your amino acids, such that UCU, UCC, UCA, and UCG all encode for serine. So this is the part where we say that we have a wobble base. So a wobble base is basically emphasizing that the third base can wobble, or meaning it can, it does not necessarily have to have a strict hydrogen bonding with the codon, or it does not have to be entirely complementary to the codon. And this is how our cell saves on its tRNA, meaning you don't necessarily have to have 4 tRNAs for serine, as in the example here. So you don't have to have 4 tRNA, one that is complementary to UCU or another for UCC or UCA or UCG. This can all be read by a single tRNA. So it's more economical for the cell if you have wobble bases. And also, this is the one that allows for silent mutations to occur, meaning even if there are certain mutations in the third base, overall, phenotypically, they do not manifest in your organism. Hence, the mutation is silent. And so you can function normally as you usually do. Here in this slide, we are just emphasizing the reading frames in that they are read 3 bases at a time. And so meaning if you have a shift in your reading frame, it will be encoding for a different type of codon or a different type of amino acid. Okay, so amino acid activation. How do we put an amino acid onto tRNA? So it's via this process we call amino acid activation, or sometimes we would also say tRNA charging. So if you see the terms a charged tRNA, it basically means that your tRNA has an amino acid. So here we look at the tertiary structure of your tRNA. And if you will remember, in our discussion on mRNA types, the tRNA has an anticodon loop here, which is the part that is complementary to your mRNA codon, and a conserved 3' end, which is largely composed of CCA. In the adenine region, this is the part where your amino acid links to your tRNA via the 3' carbon. So this is your 3' carbon. This is the carbonyl carboxyl group of your amino acid. So if you will remember, your amino acid has a central carbon, an amino group, a hydrogen, and an R group bound to it. So this is how it would look like. So the amino acid is linked to the adenine of the conserved CCA terminus in your tRNA. So amino acid activation occurs in 2 steps. The first is your amino acid is activated with ATP. So they form an amino acyl AMP plus pyrophosphate. So pyrophosphate is basically something that is released after this initial reaction. After which you have your amino acyl AMP. So this AMP here is replaced by your tRNA so that you can get amino acyl tRNA plus adenosine monophosphate. So basically, the point of your ATP reacting with amino acid is to make the amino acid activated so that it would want to interact with your tRNA. Otherwise, amino acyl tRNA will not form. Now, once you have your ATP was cleaved once in that you have your AMP plus the pyrophosphate. But this pyrophosphate also eventually degrades into 2 PIs, meaning there were a total of 2 covalent bonds rather that were broken so that your tRNA will be bound to an amino acid. So this makes the overall reaction irreversible. So the enzyme that catalyzes this reaction is an amino acyl tRNA synthetase. So when you encounter the term synthetase, it means it's something that is involved in synthesis. And if it says synthetase, that means energy was involved in this synthesis. So in this case, the energy source was ATP. So this is the mechanism for binding the amino acid to the tRNA. So if you can see, the first part wherein you are activating the activating the amino acid with ATP, it's quite general for all, but they do diverge in their in the part where the amino acid has to interact with the tRNA. So there are two types of synthetases. You have class 1 amino acyl tRNA synthetases and class 2 amino acyl tRNA synthetases. So here, this is the part where your ATP interacts with the amino acid. So this is your amino acid, this part, and this is the part of your adenosine monophosphate. So now we have an activated amino acid. And here, this is your activated amino acid, and this is the 2' hydroxyl group of the adenine found in your tRNA. So, via a nucleophilic attack, your amino acid is now linked to the 2' end of your adenine in tRNA. So in class 1 synthetases, what you produce is an amino acid bound to the 2' carbon of your adenine. Whereas in class 2 synthetases, it is the 3' end or the 3' carbon that interacts with the amino acid. So this is the final product that you want. You actually want the amino acid bound to the 3' carbon. But because in your class 1 synthetases it is bound to the 2' carbon, it still has to undergo trans-esterification so that all your products will end up with a 3' amino acid 3' amino acyl tRNA. So evolutionarily speaking, they do not know why organisms have to have a class 1 and a class 2 synthetase, but we just know that they exist in nature. So here, this gray, fleshy looking thing is actually your amino acyl tRNA synthetase, and these are very specific to your tRNA. So this violet molecule is your tRNA, and the green molecule is your amino acid. So the amino acyl tRNA synthetase, if you have a phenylalanine, a tRNA phenylalanine, there's a specific synthetase for it. So your synthetase has to be super smart such that it will recognize what type of tRNA you are so that it will know what amino acid to put into that tRNA. So if you will find this tRNA here is tRNA phenylalanine, has a different structure than the tRNA of methionine. It's also different from the tRNA for serine and the tRNA for alanine. The yellow parts here highlight the parts that are recognized by your tRNA, amino acyl tRNA synthetase. So it's wrong to think that they are recognized solely based on their anticodon, because there are also other parts in the tRNA that are recognized by the synthetase. So as you will see in this video, this is your tRNA.
And all tRNAs have a characteristic L shape, with an amino acid attached to the 3' end at the tip of the shorter arm. The anticodon loop is positioned at the opposing end of the molecule and contains the anticodon base triplet. The amino acid, a phenylalanine in this case, is covalently attached to a conserved sequence, CCA, that is common to the 3' terminus of all tRNAs. The anticodon is comprised of 3 nucleotides complementary to the codon in the mRNA. The bases are exposed and are thus freely accessible for base pairing during protein synthesis. In this example, the anticodon sequence is GAA, which would match a UUC codon on a messenger RNA, specifying phenylalanine. The charging of tRNAs with the correct amino acids is carried out by amino-acyl tRNA synthetases. As revealed in the cutaway view, the complex of phenylalanine tRNA with its cognate synthetase shows an extensive contact surface that includes recognition sites for the anticodon base triplet. The tRNAs.
Okay, so what this part is emphasizing, so if you look at the, if you cut through the amino-acyl tRNA synthetase, you will see that its interaction with the tRNA is very extensive. It recognizes this part until this part and this part, which means that it recognizes the entire surface of your tRNA. And that's how it recognizes what type of amino acid to link to your tRNA.
CCA end is deeply buried in the enzyme.
Okay, so now that we have our tRNA charged, we can proceed to translation. So we're going to start with translation in prokaryotes first. So translation occurs in ribosomes, so they are the protein synthesis centers, or they are your protein factories. Prokaryotic ribosomes are largely named as 70S, S meaning the sedimentation coefficient, or basically that's the 70S is the unit with which these were isolated. So the 70S subunit has a large subunit and a small subunit. The small subunit is named 30S, whereas the large subunit is named 50S. In the large subunit of your prokaryotes, it will contain the 23S RNA and the 5S RNA. So these are ribosomal RNAs incorporated into the 50S subunit. Whereas in the small subunit, or the 30S subunit, it will contain the 16S rRNA. So once again, we would be emphasizing that the ribosome contains both protein and RNA, but it is the RNA environment that catalyzes translation. So 30S combined with the 50S subunit produces an intact 70S subunit. So take note that these numbers are not additive. Okay? They are named based on the parameters or the situations or conditions with which they were isolated. So this is for your prokaryotic ribosomes. For the eukaryotic ribosomes, on the other hand, a whole ribosome is called 80S, whereas its small subunit is called 40S, and the large subunit is called 60S. In the 60S subunit, it will contain the 28S, the 5.85S, and the 5S rRNA, whereas the 40S subunit, or the small subunit, it will contain the 18S rRNA. So these are images of your ribosome, wherein this is a small subunit and this is the large subunit. This is their view from the side, and if you will see, there's a tunnel here. So this is where your polypeptide chain exits while we are synthesizing our protein, whereas this hole here is where the mRNA and the tRNAs are situated for translation to occur. So this is a video of your ribosome.
The crystal structure of the ribosome reveals many insights into the molecular mechanism of translation. Zooming in on the large ribosomal subunit shows highly evolutionarily conserved RNA bases lining the active site of the peptidyltransferase center, which catalyzes polypeptide bond formation. There are no ribosomal proteins in the vicinity. Peptide bond formation is catalyzed in an environment exclusively made of ribosomal RNA. The 3' end of a
Okay, so we're going to emphasize that inside the ribosome, it is the rRNA environment that allows the catalysis for the formation of peptide bonds, okay? So it's the RNA component, not the protein component, that allows the synthesis of the peptide chain.
tRNA charged with an amino acid is bound in the active site. This amino acid represents the carboxy terminal amino acid of a growing polypeptide chain on an actively translating ribosome with the peptidyl tRNA bound to the P site on the ribosome. Conserved bases common to the 3' end of all tRNAs base pair with the ribosomal RNA to position the amino acid precisely. The incoming amino acid linked to its respective tRNA binds closely, again held precisely by base pairing interactions between a conserved base on the tRNA and ribosomal RNA. A network of hydrogen bonds positions the reactive groups with the precise geometry required to catalyze peptide bond formation.
Okay. Oh, sorry. There, sorry.
The crystal structure binds closely, again held precisely by base pairing interactions.
There. Okay, so if you see the yellow parts, those are your rRNA, and the green molecule is your tRNA, and the violet one is also your tRNA. So they are found at specific sites in your ribosome, and they will have they will be undergoing a process that is specific to that position.
Between a conserved base on the tRNA and ribosomal RNA. A network of hydrogen bonds positions the reactive groups with the precise geometry required to catalyze peptide bond formation.
So if you will see here, what your ribozymes are doing is that they are making this tRNA close enough to this tRNA so that the amino acids will be so close to each other that it will catalyze the formation of a peptide bond. There you go.
The empty deacylated tRNA is released from the P site. During the translocation step of protein synthesis, the other tRNA, now containing the growing polypeptide chain, moves from the A to the P site, where it will be waiting for the next incoming amino acid to repeat the polymerization cycle. The different states of the reaction cycle shown in this animation are based on actual crystal structures, in which large ribosomal subunits were crystallized with various aminoacyl tRNA analogs bound to them, mimicking the discrete steps in the reaction cycle.
Okay, so that one was outlining the ribosome, the rRNA environment provided by the ribosome, so that peptide bond formation is enabled. In this next video, we're going to be shown how the
comparison of two states of a bacterial ribosome,
moves.
Either with the initiator FMET tRNA bound or with elongation factor EFG bound, reveals the significant conformational changes that the ribosome is thought to undergo during each elongation cycle. The ratchet-like rearrangements at the interface between the two ribosomal subunits may help move the mRNA and tRNAs through the ribosome during protein synthesis. The models shown here are a computer reconstruction made from many thousands of images of single ribosomes in vitreous ice that were observed with an electron microscope.
Okay. So when you say ratchet-like movement, it basically means that the subunits push against each other so that they can move. So this is not like a burger that you separate you separate both breads from each other and then they move at the same time. No, they move by pushing against each other. So that's like one at a time. Okay, so how do we start prokaryotic translation in your prokaryotes? So in initiation, we have to take note of three initiation factors, conveniently named IF1, IF2, and IF3, as in initiation factor 1, initiation factor 2, and initiation factor 3. So you have the 30S subunit, or the small subunit of your ribosome, and in it, initiation factor 3 binds near the E site, and initiation factor 1 binds near the A site. So that's because your ribosome has 3 sites. It has A, P, and E, wherein the A site is the acceptor site, the P site is your peptidyl site, and the E site is the exit site. So meaning all incoming tRNAs will go into the A site, and all tRNAs with an elongated polypeptide chain is found in the peptidyl site, whereas the E site is where an uncharged, or a tRNA that does not have an amino acid anymore, sits for a while before it is released. Before before the tRNA is released into the cell. So it's kind of like your pre-departure area rather. It's a pre-departure area before the tRNA leaves the ribosome. So here, your initiation factor 1 blocks the possible A site so that the initial tRNA does not bind here, whereas initiation factor 3 binds near the E site, and it prevents the large subunit from interacting with your small subunit. That's because you still want to assemble everything before the 50S subunit comes in. So the next thing that happens is your initiation factor 2, or IF2, brings the initial tRNA. So the initiation tRNA is largely called tRNAi. So while the initiation tRNA responds to AUG, which codes for methionine, this methionine here is a special methionine because it is for initiation. Hence, there is a special tRNA dedicated to it, which is your tRNAi. Specifically, in prokaryotes, the methionine is formylated, meaning there's a formyl group attached to it. So that's why sometimes you will see this initiation tRNA known as tRNA F-met. So your tRNA is brought to the 30S subunit by the initiation factor 2, whereas here you have your mRNA also brought to the 30S subunit. Your mRNA, it has a part called the Shine-Dalgarno sequence. The Shine-Dalgarno sequence is something that is conserved across your prokaryotes, and it is important in positioning the mRNA. So once your mRNA is on the 30S subunit, your tRNA, your initiation tRNA binds to it. So this will be corresponding to the P site in the 50S subunit. So altogether, you now have your 30S initiation complex. Once this is assembled, your 50S subunit can come in. So your initiation factor 3 is removed, so as your initiation factor 2 and initiation factor 1. And they are largely removed by the hydrolysis of GTP. So GTP is used up, and so these factors dissociate. So now your 50S subunit can bind successfully to the initiation complex. So together, you now have your 70S initiation complex. And now the A site, which is largely found in your large subunit, is free to position your incoming tRNAs. So this slide emphasizes your initiation codon, which is your F-met, and how it is formed largely via transferase, as in the 4-mil group is transferred from N4-mil THF or tetrahydrofolate into your methionine, so that together you can have an N4-mil methionine tRNA IF-met. This slide emphasizes the Shine-Dalgarno sequence. So the Shine-Dalgarno sequence is complementary to the 16S rRNA. If you will remember, the 16S rRNA is found in the 30S subunit of your ribosome, or it is in the small subunit. So because they are complementary, the Shine-Dalgarno sequence helps in aligning the mRNA in the small subunit. Remember that the mRNA has to be read in the proper reading frame, otherwise it may encode the wrong protein. So here, AUG is your initiation codon. Once again, AUG encodes for methionine. Okay, so this is a summary for the initiation factors needed in prokaryotes. So this is a video for translation, and apparently it's not working. Okay, we'll go next. Okay. So here we're going to go for chain elongation. So here you have your large subunit. This is your small subunit. This is the mRNA that has already been positioned. In its P site is your initiation or initiator tRNA. So now the A site is vacant. So it's ready to accept a new tRNA to come in. So in the elongation part, we have three elongation factors to take note of. We have EFTU, EFTS, and EFG. So here you will find that EFTU, elongation factor TU, is conjugated to a GTP. So together, they bring the next tRNA into position here. And once that tRNA has successfully bound to the mRNA, GTP is hydrolyzed. So meaning GTP is consumed, as in GTP becomes GDP plus BI. There. So it's consumed in the process. So now your EFTU has GDP associated to it. Now GDP is not an energy source for elongation. It's GTP. So this is where EFTS comes in. Because what EFTS does is that it replaces GDP with GTP, so that once again we can get EFTU GTP ready for another tRNA to be brought to the A site. Okay, so now let's focus on this tRNA already bound to the A site and your initiator RNA bound to the P site. So the P site are in your large ribosome, specifically the 23S rRNA, found in your large ribosome, acts as a peptidyl transferase because it transfers the amino acid from this tRNA towards this tRNA. So we read our mRNA from 5' to 3', we make our polypeptide chain from the N terminus towards the C terminus. So once this gets transferred here, this tRNA is now uncharged. This is where your EFG comes in. So EFG is bound to a GTP and it binds near the A site. So that once it binds near the A site, it facilitates translocation. So when you say translocation, it's the part where your ribosome moves. So now when your ribosome moves, this tRNA, which used to be at the A site, is now in the P site, and the tRNA that once was in the P site is now in the E site, or the exit site, where eventually this tRNA will be released into the cytosol or into the cytosolic environment. So now the A site, once it is free, it's ready to accept the next EFTU with the tRNA. So if you will notice, in translation, we are using GTP as the source of energy and not ATP. So this is a video for elongation.
To extend a growing polypeptide chain, the ribosome must select the correct amino acids that are specified by the messenger RNA. An aminoacyl tRNA bound to elongation factor TU, EFTU for short, enters the free A site on the ribosome. If the anticodon of the charged tRNA does not match the codon in the messenger RNA, the tRNA is rejected. The process of trial and error repeats until the correct tRNA is identified. Elongation factor TU hydrolyzes its bound GTP and dissociates. If the tRNA is correctly matched and remains bound for a long enough time, it is committed to be used in protein synthesis. The ribosome catalyzes the formation of the new peptide bond and undergoes a dramatic conformational change. Elongation factor G binds to the ribosome. Hydrolysis of GTP by elongation factor G switches the ribosome back to the state in which it can accept the next incoming tRNA.
So if you will notice, in translocation, the large and the small subunit, they move against each other, or they slide against each other. So now let's go to termination. So in termination, we have three release factors. So here there are several stop codons, UAA, UAG, and UGA. So these three stop codons are found on the mRNA such that once you see them, for example, here you have UAA, they do not have a tRNA specific for them. Rather, they are recognized by release factors. So we have three release factors. They are RF1, 2, and 3. So very conveniently named. So RF1 and 2 recognize the three different stop codons. And so in this example, RF1 recognizes UAA. If it recognizes UAA, RF1 binds to it, and then RF3, that is conjugated to a GTP, goes there and binds to RF1. In doing so, GTP is hydrolyzed. So once GTP is hydrolyzed, it converts the peptidyl transferase in the P site into a hydrolase, meaning now your polypeptide chain will be transferred to a water molecule instead to another tRNA. In doing so, the polypeptide chain is released into the cellular environment. And so now you have your uncharged tRNA and the release factors. So in this case, an RRF comes in. It's a RRF meaning ribosome recycling factor. It comes in and facilitates the dissociation of your tRNA as well as your release factors. So now you have an inactive 70S ribosome here. Now, for your ribosome to be able to participate in another round of translation, the small and the large subunit has to dissociate from each other so that the small subunit can once again entertain an initiation factor 1 and initiation factor 3. Otherwise, translation will not occur because the mRNA will not be able to bind with the small subunit. So, which means that after inactivating the ribosome, it has to dissociate for another round of translation. So, this is a summary of your translation in prokaryotes. So, in this figure, polysome is shown. Polysome, also known as a polyribosome. So, if you can see here, this is a single mRNA that is read by several ribosomes, and they are producing polypeptide chains at different time. It's like a staggered time. So we have a video for that.
As soon as a messenger RNA molecule is transported from the nucleus to the cytoplasm, ribosomes begin to translate the sequence into amino acids. Typically, many ribosomes translate the mRNA simultaneously. Each ribosome begins at the 5' end of the mRNA and progresses steadily towards the 3' end. New ribosomes attach to the 5' end at the same rate as the previous ones move out of the way. These multiple initiations allow the cell to make much more protein from a single message than if one ribosome had to complete the task before another could begin. When a ribosome reaches a stop codon, the ribosome and the new protein dissociate from each other and from the mRNA. This electron micrograph depicts a membrane-bound polyribosome from a eukaryotic cell.
And there we go. Now, let's go to translation in eukaryotes. So, if you will recall, in eukaryotes, we have an mRNA that has a 7-mg cap or a 7-methyl GTP cap at the 5' end and a polya tail in the 3' end. So in eukaryotes, it's usually the initiation part that is more complicated in a sense that it will require more initiation factors compared to your prokaryotes. We're not going to go into each of the initiation factors in detail, but we will be emphasizing on a few ones. So for example, you have here your 40S subunit, which is the small subunit for eukaryotes. It will require an initiation factor 1A to bind near this site, which in prokaryotes would be called the E site, but technically in eukaryotes you don't have an E site. You have an initiation factor 3 that is bound to this near the P site, and initiation factor 2 which is bound near the A site. So this initiation factors are needed so that it can set up our translation in eukaryotes. So 1A prevents the binding of your 60S subunit, which is the large subunit for your eukaryotes, whereas the initiation factor 2, or EIF2, is for bringing the initial or the initiation tRNA into position. So whenever you see an initiation factor that has an E at the start, that means this will be referring to eukaryotic initiation factors. So if you will find here, even without the mRNA, the initiation tRNA has already been bound. Unlike with the prokaryotes, you will require the mRNA before you put in the initiation tRNA. So here, when you have all these three initiation factors with the 40S subunit, you form the 43S pre-initiation complex. Now, the mRNA comes in as well as the initiation factor for complexes. So we're not going to outline each of these initiation factors. We will just call them the initiation factor for complex in general. So once you have your mRNA bound, and then it positions itself into your 40S subunit, the factors can, and once the start codon has been identified, the initiation factors can dissociate so that the 60S subunit can come in and together form the 80S initiation complex. So if you will remember in the prokaryotes, the Shine-Dalgarno sequence is important to align your mRNA with the small subunit. In eukaryotes, it is the 7-mg cap or the 7-methyl GTP cap that helps in aligning the mRNA with the 18S subunit of our small subunit in eukaryotes. So these figures are just looking through the process in a larger image, basically. And here, this is how your 48S pre-initiation complex really looks like. You have the 40S subunit here, you have the tRNA here, and you have your the other initiation factors here, and this is your initiation factor for complex. So what they do is that they use the 7-mg cap as well as the poly-A tail to kind of scan through the mRNA until it finds the AUG. So basically, they are trying to rotate this mRNA or to pull through the mRNA until you find the start codon, which will bind to your initiation tRNA. So once that is done, that's when the 60S or the large subunit comes in to form your 80S initiation complex. So this is the summary for the different initiation factors. You don't have to memorize them. You just need to focus on several of the factors, such as the initiation factor 1A and the initiation factor 2, as well as the initiation factor 4 complex. Okay. Now, in eukaryotic translation, so in our ribosome, so if this is the large subunit, so 60S, and this is a small subunit, so that's 40S, they only have an A and a P site. They do not have an E site. So somehow, when your tRNA is uncharged, it just leaves the ribosome. So here, we only have two elongation factors, EEF1 and EEF2. However, EEF1 has two subunits. They are EEF1A and EEF1B. EEF1A functions similar to EFTU, and EEF1B functions similar to EFTS, meaning it is EEF1A that brings the tRNA into the A site, and then once GTP is hydrolyzed into GDP, an inorganic phosphate, your EEF1B replaces GDP with GTP so that your EEF1A can once again bring another tRNA into the A site. EAF2, on the other hand, functions similarly with EFG, meaning their main function is for translocation. So remember that in the large subunit, it is the 25S subunit that provides the rRNA environment that catalyzes peptide bond formation. Or it is the 25S rRNA, no, the 28S rather, 28S rRNA that contains the peptidyl transferase activity. Okay. So let's now go into termination of eukaryotic translation. So in eukaryotes, termination, we have the eukaryotic release factor 1, which recognizes all the 3 stop codons. So unlike in prokaryotes, you have RF1, which recognizes UAA and UGA, whereas RF2 recognizes UGA and UAG, I think, if I'm not mistaken. So you need 2 release factors to recognize all 3 stop codons, whereas in eukaryotes, you only need 1 to recognize all 3 stop codons. And so eventually, this will facilitate the release of our polypeptide chain. Although, in eukaryotes, there is something known as the suppressor tRNA. So when this tRNA binds to the ribosome or to the mRNA, it actually disregards the stop codon, meaning translation will continue on even if there is a stop codon already. So there are certain cases where this is observed. So briefly, in eukaryotes, there are several post-translational modifications that can occur. One of them can be via cleavage. So in this example, we have pre-proinsulin, meaning precursor progenitor insulin. So this is the primary polypeptide chain, and so it will get cut here and here and here. And so once they are cut, they form the mature insulin. So trimming is one way of a post-translational modification. Sometimes you will also have glycosylation, meaning you add in sugar groups to your protein. You can also have amination, adding of amino groups, etc. So there are a lot of things that can be done to your proteins as a modification after translation. And post-translational modifications usually occur in the endoplasmic reticulum, also in the Golgi apparatus. So here is a video, briefly, of how post-translational modification can occur. So here you have your mRNA exiting the nucleus.
The signal sequence of a membrane protein directs the ribosome to the rough ER. The growing polypeptide chain enters the lumen of the ER through a...
Oh, ba yun?
The signal sequence of a membrane protein directs the ribosome to the rough ER. The growing polypeptide chain enters the lumen of...
So if you will see, this is your mRNA, this is your ribosome. And the polypeptide chain they are synthesizing has, in this case, an indicated pink component, which is your signal sequence. It's basically the sequence that directs the polypeptide chain on where to go. And so it gets trimmed off in this example.
ER through a protein import channel in the membrane. There, the signal sequence is cleaved, sugars are added, and the protein folds. Some enzymes or structural proteins remain in the ER. Most proteins that are soluble in the ER are transported in vesicles to the cis Golgi.
So here in the Golgi, if you will remember what the Golgi is for, it is for the transportation or sending the proteins either outside the cell or if you need them in your lysosomes or wherever else it is needed.
In the cisternal maturation model, the cis Golgi matures to become the medial Golgi. A new cis Golgi is formed by fusion of ER vesicles. The maturation process includes retrograde vesicular transport of resident Golgi proteins. Proteins destined for secretion after modification in the Golgi are transported from the trans Golgi to the plasma membrane. When the membranes fuse, the proteins are released into the extracellular space. Soluble proteins on the outside of a cell can re-enter by endocytosis and are sorted to lysosomes for degradation. Other proteins, including lysosomal enzymes, sort directly to lysosomes from the trans Golgi.
Okay, and so this is the end of our lecture for translation for both prokaryotes and eukaryotes, as well as for post-translational modifications. So the next lecture will be about nucleic acid techniques.